The last class we were talking about genetic code and the features of genetic code. So obviously this should be your very what do you tell aware. You're very much aware of this particular concept, if I'm not wrong, since it's definitely coming in your board examinations like CBSE 12th board examinations. What are the features of genetic code? So you would be studying about it a lot, correct? No. So anyways, so first we have what is a genetic code? A genetic code is basically a sequence of codes that is present in a functional gene. So now, how many codes make up the genetic code? Like for example, if I take, you already know that if I have a series of, for example, if I start from 5 prime end, and if I start writing code, okay, let's assume that A is C, C, U, G, C, C, U, something like this. And this is the 3 prime end of the mRNA. Now let's divide this into 3, 3. Okay. So we are dividing it into 3, 3. I mean, like this, you somewhat, I'll divide it into 3, 3. Now you can see here, each 3 letters or alphabets, they act as a genetic code. So what do they code for? Each code, so these 3 letters are one code. No, these each code codes for one amino acid. So that's what it is. Okay. So let's see. We know that there are 6 features for the genetic code, and genetic code is triplet in nature, and they are made up of the 3 specific nucleotides or 3 nucleotides. It can be any repetition. It can be U also. It can be G also. It can be AUU also. Okay. So it's not specific that whether the code should have no repetition or repetition, something like that. No, any 3 series of alphabets, especially within AUCG. Within AUCG, they can combine to form a genetic code. Okay. So genetic code is universal. So this is very important concept. So genetic code is universal in the sense from bacteria all the way to the complex organisms. You take any organisms, if they have genetic material and if they have RNA. Okay. So in RNA of bacteria and in RNA of humans. Okay. This is bacteria. This is human beings. Okay. So if I take AUG, the code AUG in bacteria, or in humans, the code AUG, both of them codes for methionine. Okay. So both of them code for what? Methionine. So this is the universality. If AUG codes for methionine, it is the same for you, a bacteria, an amoeba, a dog, etc. So that's what is called as universality of the genetic code. Okay. Next one. So they are not overlapping, and they share the nucleotides. Overlapping in the sense, basically you don't have the separations, and it is not having any of the denotions like comma, full stop, exclamation, nothing. Once you start with 5 prime end, you end at the 3 prime end. And the code is degenerative, which is such an important nature of the genetic code. What is degenerate codes? Okay, more than one code can code for a single amino acid. Okay, we have a degenerate codes here. Let's see what are degenerate codes. Can you please spot me some degenerate codes? For example, let's say here we have CUU is one code, and then you have CUC is another code, and then we have CUA is another code. You can see that there are different codes coding for the same amino acids. That means more than one code can code for the same amino acid. So this is called as a degenerate nature of the genetic code. You understand my point, guys? So most of the degenerate codes, what you have is the first 2 letters or the alphabets are same. You can see here in this case, G. So they are same. Whereas the last alphabets are only different. You can see UCAC. You understand my point? So it's very easy to understand. I mean, like recognize a degenerate codes if they are given in a series. So for example, A. You see the last letters, the last alphabets are different. So they are called as degenerate codes, and the code is degenerative nature. So in total, we have 64 codes. Out of 64 codes, 61 codes for amino acids, correct? No? And 3 of the codes do not code for any amino acids. And these 3 codes are called as nonsense codes. Why do they call as nonsense codes? Because there is no specific amino acids are assigned to these. Okay, so that is why they are called as nonsense codes. What is your nonsense codes? Usually I pronounce it something like this. Ua, I will write it here. Okay. Ua, uag, and uga. So UAA, UAG, and UGA are the three codes which do not code for any amino acids. So probably you guys are very much aware of the nature of the genetic code, the discovery of the genetic code, and the triplet nature of genetic code, 64 codes, etc. Okay. Now, why did we study genetic codes? We studied genetic codes because we understand that the DNA was converted into an mRNA, and through mRNA, we are supposed to synthesize something called as proteins. So, what are proteins? Proteins are made up of protein is a polymer. It can be any type of polymer. It's basically a heteropolymer. So the number of chains making up a protein can vary. A protein can be a single polypeptide chain. It can be a combination of two polypeptide chains. It can be a combination of two similar kinds of a polypeptide chain, etc. So based on that, we have categorized the proteins as primary proteins, secondary proteins, tertiary proteins, and quaternary proteins. Correct? No? In your first year, you might have studied that protein structure can be primary, secondary, tertiary, and quaternary structures. That we have understood. Now, coming to the most basic thing on how what makes up the protein. The proteins are made up of what, guys? It is made up of amino acids. It is made up of what? Amino acids. So if you see the polypeptide chain, a polypeptide chain contains a series of amino acids that are adjacent to each other. So if you start at one end of a protein, and if you keep on looking at what is this protein made up of, there is one amino acid, there is another amino acid, and another amino acid. All of them are arranged in adjacent to each other. How are they arranged? They are not just arranged, they are attached to each other by a formation of a peptide bond. Okay? A peptide linkage, a type of covalent bond. A peptide linkage is established from one amino acid to another amino acid. Now, the question is not how they are linked. The question is how they are arranged. Correct? No? If you see, if I take uracil, okay, or if I take, sorry, if I take valine, let's say, if I take valine, if I take glutamic acid, if I take one more, leucine, isoleucine, okay, isoleucine. This is just like, okay, vaguely I'm writing. So this is all amino acids, guys. Now let's assume that here there is valine, here there is valine, here there is glutamic acid, here there is leucine, here it is isoleucine. Who really told the machinery? Correct? No, translation is a machinery. So translation is a process, and translation machinery will be there. No, so the translation machinery means anything that is involved in the process of translation is called as translation machinery. So how does translation machinery really know what amino acid to arrange next to what? Which amino acid should come next? So that is determined by the arrangement of genetic codes. So if you have genetic codes, like for example, U, A, C, G, G, A, C, G, A, UU. Okay, so these are our codes, guys. So this is just a mRNA. Now, how does this mRNA behave? Here, this will code for one amino acid, and this will code for another amino acid. This will code for another amino acid. This will code for another amino acid. Similarly, like it goes on like that. Now, UAA code for one amino acid, or it might be the same amino acid here. We don't know. Okay, so it knows this code will code for this particular amino acid and it will bring it here. Next, this code will code for another amino acid and it will bring next to here and it will create a bond like this. So proper arrangement of amino acid is what makes a specific protein. Now we might have studied, we studied actually basically, while we were discussing about principles of inheritance and variation, we saw a particular mutation called as point mutation. Correct, no guys? Now, what is the example for point mutation? Can you give me? What is our example for point mutation? Come on, guys. What is our example? What's the example for point mutation? It's a very general question. Do you have an answer? Or are you able to hear me, guys? Okay. Anyways, like while you answer, I'll just move with the process. Okay. So the arrangement of amino acids is very important. Why arrangement of amino acids is very important? You know that not every amino acid is same. Our amino acids are what? Some of them are polar, they are non-polar, some are positively charged, some are negatively charged, some are aromatic. Okay, so the amino acid nature changes, their charges changes, the interaction changes. So based on what amino acids are coming with beside each other, the nature of protein also determines, the structure of protein also determine. Correct? No, in point mutation when we study sickle cell anemia, just removal of one amino acid. Correct? No. So instead of glutamic acid, valine was replaced. Just because of that, now what happened? The entire structure of hemoglobin changed. Correct? No. So that is why. So this is such an important process. Genetic codes are such important codes. So now what we're going to study is in translation. How are the translation machinery completes this process of construction of a polypeptide chain. Okay. So if you see the translation, there are 5 steps basically in the translation. First one is we need to make sure that we have amino acids coming up for the site, like for how the bricks come for the construction site of a home. Similarly, the amino acids have to be assembled. Somebody has to bring us the amino acids. Correct? Next one. Once you have the amino acids, so these amino acids have to be carried by someone. We need a vehicle. So these amino acids are carried by something called as tRNA, a transfer RNA. So the transverse RNA or the transfer RNA, they carry these amino acids through a process of activation. I'll talk about how the amino acids are activated and how they get attached to the tRNA. Got it? Next one is initiation of polypeptide chain. So for initiation of polypeptide chains, that means we are talking about initiation of translation. To initiate a polypeptide chain, what is required? The translation machinery is required. Who is involved in translation? rRNA is involved in translation, ribosomes are involved in translation, mRNA is involved in translation. All have to come at one place and all have to play a one role. So they come, they attach and recognize an mRNA, and then this initiation is complete. Once the initiation is complete, now what do? We go for elongation of polypeptide chain. And finally, it comes with termination of polypeptide chain. So these are the steps that is involved in the process of your translation. First, we talk about the translation of First we talk about the activation of your tRNA. What is called as activation of tRNA? Now, let's say that we have amino acid. Okay, we have amino acid, and amino acid basically it reacts with ATP. Amino acid reacts with what? Adenosine triphosphate. So to react with adenosine triphosphate, an enzyme called as amino acyl synthetase. Amino acyl synthetase. So this is a very important enzyme, amino acyl synthetase is an enzyme. So in the presence of amino acyl synthetase, what will happen is, okay, this enzyme will actually proceed in the presence of Mg2 plus, very important equation, okay, or very important concept it is. You have amino acid, it reacts with ATP. Once it reacts with ATP, for it to react with the ATP, there is an enzyme called as amino acyl synthetase. So this amino acyl synthetase will react in the presence of magnesium. So what will happen here? We have So we have amino acid plus ADP, AMP. Okay, amino acid plus AMP, it will form. Okay, this will form amino. I will not write plus. I will write it as amino acid AMP will be formed. So this complex is formed with an enzyme called as amino acyl synthetase. So once this amino acyl synthetase is done, now what will happen? A specific tRNA comes. Okay. So this process of creating amino acid and AMP complex is called as activated amino acid. So this amino acid and AMP, they will attach to specific sites of the tRNA. Okay, so this attachment of an activated amino acid to the tRNA is called as activated tRNA. Okay, this is called as what? Activated tRNA. So how do they get the energy? So this AMP is the one that is used for the construction of bond between tRNA and amino acid. So where does the amino acid attach to the tRNA? We know that at the amino acid binding region. Okay, we have amino acid binding region in the tRNA, right? So that is where it will attach. So what is the process? What did we understand? We have ATP, we have amino acid. They both combine to form what? Amino acid plus AMP. Okay, amino acid and AMP. Amino acid and AMP. So this process is brought about by an enzyme called as amino acyl synthetase. That is done. So once we have this, okay, this will go and attach to the tRNA. Now your question is, sir, where do we get the energy from to attach the amino acid to the tRNA? We already have an AMP molecule. The AMP molecule will release the energy when it breaks the bond between amino acid and AMP. So this energy is utilized for the attachment of amino acid to the tRNA. Simply does not attach, no. A specific amino acid has to be recognized. So the DHU loop, as we talked about it in the last class, the role of DHU loop is to recognize specific activated amino acid. How does it recognize a specific amino acid, activated amino acid? Let's say that we have tRNA, and in the tRNA, we have something called as anticodon loop. What is this anticodon loop? A code is something present on the mRNA, and anticodon is the code that is opposite to this code. Let's say that it is UGC or something like that. Okay, so this is a code. So this is an anticode. Based on this anticode, a DHU loop will recognize what amino acid is this amino acid this code will code for. So it brings about this amino acid, and it will make sure that the tRNA carries that specific amino acid. So once you have your activated amino acid being recognized by your DHU loop, it goes and binds to what? It goes and binds to the 3' end of your tRNA. So once it goes and binds to that tRNA, now what we will call? We call it as amino acyl tRNA. We call it as what tRNA? Amino acyl tRNA. Why we call it as amino acyl tRNA? Because it's carrying the amino acid. Clear, guys? So now we have the source to bring amino acid. Now we see what is happening with respect to the translation process. If you look at the site where the translation has to happen, it has to happen at the mRNA. mRNA is where the messages are. So that is where everything should assemble, like how it happened in DNA. Here, guys, we have something called as an mRNA, and mRNA, we have 5' end and we have the 3' end. Now, if you see your prokaryotes and then you have your eukaryotes. Now we know that in our eukaryotes, there was one process called as capping, correct? No, we had a 5' cap. So we added a 5' cap because why? This 5' cap that is present acts as a recognition site. So this cap is recognized by who? It is recognized by the ribosome. Okay, so now we go back here. We have something called as ribosomes. What is the ribosome? A ribosome is a combination of RNA and protein. Okay, it is a macromolecule. A ribosome is what? It's a macromolecule. We know that the ribosome is made up of two subunits. It is small subunit and large subunit, correct? No, or big subunit. The small subunit, what it will do is it will take the help of rRNA. The rRNA will attach to the small subunit. So when it attaches to the small subunit, rRNA will give the information to the small subunit what RNA it is. So this will help our small subunit to come and attach the cap region. So it will recognize the cap region. So once it recognizes the cap region, the small subunit will come and sit in the 5' end. Clear, no? And next, what will happen? Following your small subunit, a large subunit will also assemble. Now the ribosome the small subunit of ribosome, the large subunit of ribosome, as well as your mRNA has recognized. I mean, like has come and assembled. So once they have assembled, this is called as what? This is, oh, sorry, I will write it here only. I don't have space. 1 second, guys. This is called as what? Polysome. Okay, 1 second. This is called as what? A polysome. What is a polysome? A polysome is a RNA plus ribosome. Okay, this complex, an RNA along with the ribosome is called as a polysome. So this is created. Clear now, guys? What did I tell? A 5' cap is recognized by your small subunit. How does it recognize the small? How does a small subunit recognize this 5' end? It will recognize with the help of rRNA. And rRNA goes and attaches to your small subunit, and it will bring the ribosome to the 5' end. So once the small subunit comes and recognizes the mRNA, a large subunit will also come and assemble. So once this assembly takes place, so this will create a polysome complex. It is also called as what? A polysome complex. Clear? No? Okay. Next, what will happen, guys? If you see the initiator codon, we call it as something called as initiator codon. What is our initiator codon? The first code that is present in an mRNA, okay, especially in prokaryotes. Okay, correct? No, we have to bring the amino acid. No, this has to lead to translation. So first we understood how the assembly has taken place. Now we have to talk about initiation. In prokaryotes, what happens? Every mRNA, it starts with a code called as AUG. So if you see what AUG codes for something called as methionine. Okay, it codes for what? Methionine. So if it codes for methionine, it has to have a dual function. So in the genetic codes, you studied that methionine has two roles. One, it acts as an initiator codon, and it also codes for methionine. Correct? No? What is an initiator codon? An initiator codon is something, see, for what happens is, if AUG is present in the beginning, okay, it codes for something called as methionine. Now, this methionine is not normal methionine. In prokaryotes, this methionine is formylated. Okay? Formylated methionine. What is the meaning of formylated? A formalin group. A formalin group, okay, attaches to this methionine. So, we call it as formylated methionine. The formylated methionine is something that acts as, the formylated methionine is something that acts as your initiator codon in prokaryotes. Understood? So if the code is AUG in the beginning, it will be not a normal methionine. It will be METF, we call it as. What is that? It's a formylated methionine that comes and sits. Okay, that is why AUG is called as initiator codon. If that same AUG comes anywhere in between your genetic code, for example, somewhere here, somewhere in between, okay, if it comes somewhere in between, so this is called as what? It is called as a normal methionine. That means it will bring a normal methionine molecule. So this exclusively happens in prokaryotes, not in eukaryotes. Why? It requires an initiator codon, correct? No? So it requires like a cap, like how we have a 5' cap in our prokaryotes. Similarly, oh God, 1 second. Excuse me, guys. Okay. So like how we have prokaryotes, similarly, even in our eukaryotes, okay. Excuse me.